A very good morning. So after such a wonderful presentation of Dr. Bikrant, I'll be changing the gears and talking about the role of edible exosomes and liver diseases. So the outline of my talk will be what are actually edible exosomes, what they carry, composition, why edible exosomes, what merits it has, isolation and its characterization, what is the clinical utility of edible exosomes in liver diseases, and edible exosomes at ILBS. So from last 2 decades, actually exosomes have drawn our attention, especially in medicine, and initially it was referred as dust particles. But now it is so very well established and has detailed about what exactly exosomes are. They are part of extracellular vesicles, the bigger umbrella, and they are cell-derived membrane-bound particles. So each cell of human, they release these extracellular vesicles. These are very small in size, nano size, but they carry a lot of information in terms of cytokines, membrane proteins, metabolites, microRNA, even DNA. And they are actually the cellular messengers. They go and crosstalk with the naive cells or the neighboring cells it has. So even in liver diseases, they've been utilized to communicate with the neighboring cells or understand the pathobiology of the diseases. They are used as clinical biomarkers, as liquid biopsies, and also been known for its targeted delivery vehicles. So the exosomes are actually not limited to mammalian cells. Even the plant kingdom, they also release their exosomes, which we are referring to as edible exosomes. And why plant releases exosomes? In normal physiology, they are released as their defense mechanism. But they have their active compounds, which are intranuclear delivery system, and which is used for them to have an innate bioactive compounds. And now they can be utilized as therapeutic delivery system, as they have the ability to have the activity even in the digestive enzymes. So these are naturally occurring vesicles which are derived from plants. They are nano size, they have higher bioavailability, they are very specific, they are innocuous, non-immunogenic, just like food. Slightly negative charge make them more potentiate that they do not make clump and helps in the extended circulation. So their physiological stability even in the GI tract under varying pH and temperature. So this is an electron microscope. Here you can see that microvesicular bodies inside the cell, they are releasing the exosomes, which are carrying all the information present inside the cell. So why edible exosomes? We can always eat fruits and vegetables and have the nutrient value, but nutrition is one of the major aspect, and it has been very much looked into liver disease, that many patients undergo malnutrition. There's an aggravated diminished appetite, exaggerated catabolic state, and loss of functions of hepatocytes. And depending on the severity of liver disease, individuals may need to adjust their caloric intake. So there's a difficulty for nutritionists to have a calorie because they can only adjust to a certain level. So what is the solution? So maybe the edible exosomes can be a solution for it. So they have very beautifully shown in initial papers that 100 microgram of ginger can act at 10 microliter of ginger exosomes. So this tissue and these small little tiny amount of ginger exosome can have equal amount of yield in terms of its protein, RNA, and also the number is so low. So we can have a ginger exosome in a very smaller quantity rather than having the whole ginger. So what exactly these exosomes carry? So they are in nano size. They carry lipids, proteins, their small RNA, and even the active ingredients. This is an example of ginger itself. So chagol or gingerol are the active compounds. We all know that it has a nutrient value, but these small pockets carry a larger amount of these active compounds. And the lipids which are present on its surface are actually different in all the different exosomes, and depending on their lipid present on the surface, they are being engulfed by the cells. For example, hepatocytes will engulf the certain type of exosomes only, whereas bacteria engulf more of phosphatidyl acid or DGCG present lipids on the exosomes. So there are many cargoes which has been defined so far, and we have tabulated it that ginger, garlic, lemon, strawberry, broccoli, grapefruit, and even the soya bean, they carry certain kind of microRNA, their active compounds like chagol, there's vitamin C, MERS 166, 168, naringenin. So we know that these compounds has a varying effect which has been studied so far. So, but how do we isolate? It's very simple. According to Food and Agriculture Organization, fruits, vegetables, and even milk has been categorized. We just do the grinding in a mixer, and first the initial low velocity centrifugation, followed by high velocity ultracentrifugation. And by using a sucrose gradient ultracentrifugation or ultrafiltration or polymer based precipitation, we can isolate these exosomes within 5 hours. So this is a kind of layers we get after isolation, which can be enumerated using an electron microscope. And this is from grapefruit, ginger, and carrot. So what unique quality they found out was though they are of nano size, but their structure is little different in each exosomes. So carrots are more of a cup shaped, ginger are more spherical in shape. So this has been known, and we can enumerate using electron microscopy, which is a very clear cut definition of exosomes. But we can also know their number and their size and concentration at the same time using a nanoparticle tracking assay. So what exactly it does is nano-sized particles usually move under Brownian motion due to the random movement of water molecules surrounding them. The smaller the particle is, the more rapid the Brownian motion, and the velocity of the motion is captured by the nanosite. So this is a video which is the movement of these small particles, and according to the movement of these small particles, the velocity is captured, and it is attached with the microscope and a laser. The refractory index actually showed us what is the concentration of particles per ml and what is the size we are getting in our fluid. So not only this, like every cell has a charge, similar way exosomes are not inert. They also carry a charge on top of it. So the more negative charge from minus 50 millivolts to 0 degree, it shows that they have a tendency of not aggregating with each other. They are more stable, and they are biologically active. They mutually repel. So this zeta potential actually tells us whether it is inert or it is more stable and it is biologically active. So we get the result of their activity. So what is the clinical utility of edible exosomes in liver diseases? So interestingly, this is one of the earliest papers in 2018 where they took healthy volunteers and they make them eat ginger exosomes for 7 days, 0 to 7 days, and then they did the stool microbiota index, and they found that within 7 days the ginger exosomes were able to change the gut microbiota. And by 16S metagenomics, they showed that lactobacillus and even the their abundance increased, whereas Clostridium and Rhamnococci level, they all decreased within 7 days. Not only this, they labeled these ginger exosomes, and they found out that they were uptaken by bacteria present in the microbiome. So they found Lactobacilli taxa was able to uptake these bacteria, and within these lactobacillus, the genes which were involved, such as LEX-A, yesinase, paxi, they all were the genes which were upregulated, and these are the genes which are involved in the differentiation of these bacteria. So we eat ginger exosomes, they are taken by bacteria, and the differentiation of lactobacillus increased. So they did it in a colitis animal model. When they gave the ginger exosomes, they found out within 24 hours the survival rate increased. So this is another paper by lemon exosomes, where they also found out that it's not only the ginger, it's the lemon which has also been taken by Lactobacillus rhamnococcus GG and STG in C. diff mortality. And these lemon exosome increased the bile resistance and gut survivability. And when they did the metabolomics, they found out that indole 3 lactic acid was involved, which was inducing IL-22 production and lactic acid. So it decreased the C. diff fecal shedding by inhibiting the growth and indole biosynthesis. So this is a science study in 2023, that not only lemon or fruits and vegetables, even the milk-derived extracellular vesicles protect the intestinal barrier integrity in NASH model. So they have taken the NASH model and given the bovine milk extracellular vesicles. They found that ZO1 and even the Claudin 1, the tight junctions present on the epithelial cells, this is the after giving the milk EVs, they found the expression increased. Not only the tight junction expression increased, but also the endotoxin levels were decreased in feces, liver, and plasma. So they found even in the mechanism that AMPK and IGF and GLP-1, the milk exosome increased the barrier integrity. So oral administration of milk EVs restores the gut barrier integrity and prevents the endotoxin translocation into the liver. So I have tabulated all the studies which has been done in terms of NAFLD, the pomegranate, garlic, blueberry, turmeric, nuts, and orange juice recently. They all found out that they are protective and are involved in various mechanisms. So the paucity of time, I am just tabulating and stimulating you that there are many studies which are done in vivo in an animal model system. So as for even for the leaky gut and the alcoholic liver disease, in vitro colitis, they are known that they are involved in the various cellular processes. So how do we translate edible exosomes from bench to bedside? So I believe edible exosomes are safe, and there will be an accelerated version from clinical trial 1 to 3. So edible exosomes, since they are safe and they are the isolation and purification of exosomes and manufacturing process is easier, that it can be scaled up and meet up the current manufacturing practices. So the regulatory approval could be simpler for this. So far, because since it is only 3 or 4 years, they are there. So there are only 4 clinical trials which are there for grape, for aloe vera and ginger, curcumin, and ginger curcumin together. So they are giving it as a dietary supplements and even in the tablet form in for oral mucositis and even for colon cancer and IBD. So edible exosomes, naturally occurring nanoparticles, they can improve the solubility of the natural compounds. They can target the natural products to a specific organ. The selectivity, drug delivery, efficacy, and the safety thereby reduces the dose and increases the patient compliance. So they are able to deliver high concentration of drugs to diseases sites because of their unique size and high loading capacities. Delivering the drug in a small particle size actually enhances the entire surface area of the drug. So therefore, the edible exosomes can be utilized for drug delivery systems, and they show an enhanced permeation and retention effect without any or less side effects. So they can be delivered in a bioengineered form. The challenges usually with exosomes is that the scalability, low reproducibility of isolation techniques, or high heterogeneity. So the solution is we can always modify, or we can put our drugs in these edible exosomes and deliver it to the patients. So there are many studies which they are understanding that they can utilize for doxorubicin or HSP or other compounds. They can be delivered using the bioengineered or modified exosomes. So edible exosomes at ILBS. So I will begin that whenever there is a liver injury, we all know there is a gut dysbiosis, and which is one of the hallmark features, and there is a change in microbiome flora. The so-called good bacteria, which converts the primary bile acids into the secondary bile acids, become less abundant. And due to the infection status, the usage of antibiotics also modulates the gut microbiome. So there is an elevated levels of urease producing bacteria, higher deconjugation occurs, which is taken up by the liver and adds on to the ammonia levels, which gets into the systemic circulation, disrupts the blood-brain barrier, and induces neuroinflammation, edema, or cognitive disorder, which leads to hepatic encephalopathy. So dietary fiber containing fermentable carbohydrates is known that these fibers can be fermented by the gut microbiome to short-chain fatty acids, and these prebiotics can be described as selectively fermenting ingredients capable of inducing specific changes in the composition. So for this, we can utilize our edible exosomes. So whenever there is a hepatic injury, there is a higher level of ammonia, and for this we are using the standard medical therapy as lactulose, rifaximin, Lola, but this all comes with the baggage of side effects. So edible exosomes, does it has any impact and can prove a novel prebiotic therapy for hyperammonia condition or not? So that was our question. So for this we took carrot, ginger, garlic, and turmeric associated exosomes, and we developed an in-house method to isolate these edible exosomes. So this is an electron microscope. You can clearly see that they are cup-shaped spherical exosomes of size range between 50 to 100 nanometer in size, and their concentration came out to be enough to be delivered to the mouse model, and they are active in terms of zeta potential. So whether they are biologically stable, so we have, the most of the work is done by Debishri in my lab. So they have done is stomach-like solution and intestine-like solution, and passed these exosomes through this solution, and found out that even after 60 minutes of their treatment with this solution, they are stable in their biological activity as well as in their size. So for this we have used a thioacetamide hyperammonia rat model. The thioacetamide was given for 10 days, and the daily dosing of edible exosomes was done for 10 to the power 11 exosomes every day. And we had 4 different groups, one thioacetamide alone, one with the edible exosomes, and as a control we used lactulose. So this is the intravital microscopy, which we saw that within 6 hours, these edible exosomes were actually labeled with PKH67 dye, and after the oral gavage of edible exosomes, they were were settling down and reaches to the liver 6 hours, after the 6 hours. So when you compare the carrot, ginger, garlic, turmeric exosomes, we saw in comparison to thioacetamide alone, the carrot exosomes showed a very effective lowering of ammonia. And these carrot exosomes comes from Daucus carota species, Pusa aceta. This is a special black carrot which we procured from IARI, New Delhi. And among all, since we got the carrot exosomes, we took it further. So these nutritional composition actually showed us that they are high content of anthocyanin and vitamin C and lycopene, vitamin K. And this is the different metabolites or groups we found out. And when we looked at the other hepatic injury markers, we found that even the bilirubin was significantly low, and so as the ALT levels, and the albumin levels were high. So when we looked at the liver histology, you will appreciate the information that in comparison to lactulose or TA, there was less number of cellular cholestasis, portal expansion, or even the necrosis after we give the carrot exosomes. So in the liver, we looked at different gene expression markers associated with bile acid receptors, inflammation, apoptosis, oxidative stress, nitrogen metabolism, and among that we found that sphingosine 1 phosphate receptor 2, which is also a bile acid receptor, was upregulated after giving the carrot exosomes, and even the nitrogen metabolism, the OTC, which is ornithine transcarbamylase, the rate limiting enzyme, was found to be higher after giving the carrot exosomes. Then we were very much interested whether these carrot exosomes changes the gut microbiota. So we took the stool samples and did the 16S metagenomics, and we found that there was no difference in the alpha diversity, but there was a significant difference in the beta diversity after giving the carrot exosomes. To much of our interest, when we compared the control, TA, lactulose, and carrot exosomes gut microbiota, we found that Escherichia shigella, which was absolutely absent in controls, there was high abundance of Escherichia shigella in the disease model, which was almost gone after giving the carrot exosomes. And the firmicutes, lactobacillus, lactobacillus, which were known to be the good bacteria, the abundance was higher in comparison to the TA after giving the carrot exosomes. So hepatic encephalopathy, we already know that there is a Escherichia shigella, the abundance is higher. So it is in the patient, which actually coincides with our rat data. So not only this, we wanted to understand whether this good bacteria, the bacteria can uptake these carrot exosomes or not. So we found that yes, after giving the carrot exosome to the LGG in the culture, the growth of the LGG bacterial growth was higher, and in the gene level, the secondary the genes which are involved in converting primary bile acids to the secondary bile acids, they were upregulated after giving carrot exosomes in the bacteria. So the metabolite signature, when we looked in among the group, we also found that the gut bacterial changes correlate with the secondary bile acids post carrot exosome consumption, and we found that most of the metabolites which are involved in converting primary bile acids to secondary bile acids were upregulated after giving the carrot exosomes. When we looked at the cognitive behavior, since it was hepatic encephalopathy or hyperamonic model, we found that even in the brain, after giving the carrot exosome in comparison to lactulose or thiacetamide alone, there was less of edema found in the brain. And if you notice this video of the rat of the tail suspension, I think I'm not able to play the video, but that's okay. We can see here that tail suspension and grip strength among the groups, we found that it was improved after giving the carrot exosomes, and there was less of inflammation in the brain. So to summarize, that carrot exosomes reduce plasma ammonia levels and improve the liver function and cognition. It inhibits the colonization of Escherichia shigella and reduce the urease producing bacteria. They potentiate a therapeutic candidates for hepatic dysfunction and gut dysbiosis and hepatic encephalopathy. So for the future perspective, edible exosomes can be considered as functional foods to promote the specific physiological functions, can be considered for drug delivery system because they are safe and they can be engineered, they can be scalable, and it can be utilized for personalized nutrition and maybe tailor-made. Microbiome modulators can be replaced by using different gut microbiome targeted therapies in liver diseases, which will be based on edible exosomes. The regulatory agencies may need to establish guidelines and safety assessment to ensure the responsible use of these products. Thank you so much for your attention.